Hello, everyone. My name is Max Vargas, and I am a fifth-year neuroscience graduate student at UC Davis, working in the lab with David Olson, where we research psychedelics and the mechanisms through which they promote an antidepressant response, as well as neuroplasticity. So what really makes psychedelics stand out is that they can promote a rapid and enduring antidepressant response. And this is in stark contrast to traditional antidepressants, where patients typically have to take the drug for weeks on end before feeling any type of effect. One of the ways that we study how psychedelics can promote neuroplasticity, or the ability for neurons in your brain to rewire, is by looking at how the structure of the neurons changes in response to treatment with drugs. So here in this panel, you can see 3 neurons that were cultured and then treated with 3 different conditions: vehicle, 5-HT or serotonin, and then DMT, a classical psychedelic. And what's really apparent is that the DMT-treated neuron experienced a lot of growth compared to the other 2. And this is really interesting once you examine this in the context of the localization of the serotonin 2A receptor, or the principal receptor that these drugs are believed to bind to and then promote neuroplasticity and the antidepressant response thereafter. And what we can see here in this panel is cultured cortical neuron, and in green, the membrane is stained. And then next to it, we can see the expression of the 2A receptor in blue. And what's interesting is that the serotonin 2A receptor is primarily expressed on the inside of the cell, as opposed to many other GPCRs, which line the plasma membrane on the outside of the cell. And serotonin, being a membrane impermeable compound, so unable to cross this membrane, it does not have access to this pool of intracellular receptors, compared to DMT, which can cross the membrane and gain access to these receptors. So to test the hypothesis that if we can get serotonin onto the inside of the cell, that it would have the same response as DMT, we performed an experiment where we expressed the serotonin transporter, or SERT, on cortical neurons. And the serotonin transporter essentially acts as a pump for serotonin to move it from the outside, the extracellular space, onto the intracellular space, thereby the idea being that it would give serotonin access to this intracellular pool of serotonin 2A receptors. So we did this experiment, and really interestingly, you can see in this middle column with serotonin, the certain negative neurons in blue that were treated with serotonin, as you would expect, didn't have much of a response. But the neurons that do express SERT and were treated with serotonin showed an increase in growth compared to the vehicle control, demonstrating that if you can get serotonin inside the cell, it'll have the same type of response as DMT, presumably because you're giving it access to this pool of intracellular 2A receptors and allowing it to cause these changes in neuroplasticity. And this type of information is really important for further understanding the way that psychedelics can promote neuroplasticity and ultimately cause an antidepressant response, and also in developing future analogs of psychedelics, making sure that they will be membrane permeable and have access to these receptors. And with that, I'd like to thank you for your time and listening.